If you don't know anybody who has cancer or has had cancer, then you are pretty lucky. Cancer cells are going to start out as normal body cells. They undergo genetic mutations, and then they lose the ability to control the tempo of their own division. So they are going to do cell division uncontrollably. They go crazy, they go wild by doing cell division over and over again, and then that can cause diseases. And so cancer therapies are going to seek to disrupt one or more of the steps in cell division, and so understanding how cell division occurs becomes very important. So then if you know how it should happen normally, then you can put in steps and come up with therapies to sort of figure out how to stop cancer in its tracks. So cell division is also very important because it's a key characteristic of life. Okay, being able to reproduce is that characteristic, and we talked about that way in the beginning of the year. It's also part of the cell theory. We said that cells come from pre-existing cells, and so how that all comes to play is going to be very important, and we'll learn about how all that happens in this chapter. Cell division occurs at the cellular level. We need to take that DNA, which is going to be in the form of chromosomes, and we need to duplicate that. We talked about that earlier as well. DNA replication, so that when we make two new cells, each cell is going to have its own set of DNA. And so in this process of cell division, we're going to have to sort out all those chromosomes so that each cell can get its own set of DNA. There are two types of reproduction. One is called asexual reproduction. It's going to produce offspring that are identical to the original parent, so identical. And it's also only going to involve one parent. Usually asexual reproduction is going to be when we talk about mitosis. Okay, and then sexual reproduction is going to involve a special type of cell division called meiosis, because meiosis is going to cut the chromosome number in half so that when both parents come together in that blessed event and join their sperm and egg, which was made by meiosis, that's when you get a whole unique offspring. And so since you're combining two different parents together, they're going to be similar to the parents, but they're not going to be identical. For mitosis, okay, which again was going to be asexual reproduction, some single-celled organisms, that's just how they're going to reproduce. For us who are multicellular, we need to do it so that we can grow, right? So when we were a fertilized egg until you are where you are right now in your life, and then continuing on into adulthood, in order for you to grow, you need to do mitosis. If you get cut, like you fall down, scrape your knee, you need to repair yourself, and so you're going to do mitosis. You need to replace cells, so all that dry skin that you have and skin cells flaking off, you need to replace those cells, and so you're going to do mitosis. Okay, so anytime you put a band-aid on, you can think, oh boy, now my body's going to do mitosis and it's going to heal itself and repair. There's also cell division, again, that's meiosis, which is going to be involved in sexual reproduction. But meiosis is the process that's going to make the sperm and the egg, okay? And again, this can happen, once you hit puberty, you're making sperm and eggs. But these things should not join together until you are married. All right, for prokaryotic cells, how they divide, it's going to be in a process called binary fission. Again, if we're learning about how lots of things divide, we should figure out how bacteria do it as well. And so they have, prokaryotic cells, their chromosomes are circular, okay, so their DNA is circular. There's some proteins involved, but usually much smaller because smaller than eukaryotes themselves. So here's a picture of a bacteria, and step number 1, the duplication of the chromosome and the separation of its copies. Okay, so DNA replication is going to happen. Remember, it is not in a nucleus because it's a bacteria here, so it's just the nucleoid region, but we still need to, okay, so we'll rewrite that step 1. Step 2 is going to elongate, so the cell is going to get longer. And the two copies, so here's like copy number 1 of the DNA and copy number 2, they're going to move to opposite ends of the cell. Notice it's starting to pinch in here. Then we'll keep repeating 1, 2, and 3. It's actually going to then divide. So the pinching will go all the way through, and we have 2 cells. Okay, so 2 cells. Where up here we just had one that we started out with identical DNA inside. So binary fission done in prokaryotic cells. In eukaryotic cells, things are going to be much more complex because we are bigger. We have more genes, and we're going to store most of those genes on multiple chromosomes within the nucleus. Different species are going to have different numbers of chromosomes. For humans, our total number of chromosomes is 46. That's important to remember. And in case we haven't mentioned it yet, genes are sections of your DNA that code for a certain trait. So that little part of the DNA that's going to code for whatever trait that we have. We might have mentioned it before. But so when we talk about chromosomes, we'll see pictures here in a second, but chromatin is what I often say it's thin. So it's going to be the DNA that's in the thin version. Okay, so it's the DNA, and it's going to consist of some proteins that will help with the structure and the control of the activity of its genes. But when you start to divide, that's like moving time, so you need to pack things. Like when you're moving your house, you would put things into boxes and you would pack things so that it's easier to move. And the same thing for the cell. It's going to take this thin chromatin and it's going to pack it all together. So it's going to be nice and condensed, compacted, so much so that we can actually see that under the microscope. So here's some lovely pictures. So when we have our DNA and these histone proteins together, that's what we call chromatin, and it's very thin and invisible. That's an I. And it's invisible under the microscope. Okay, so when the cell is in chromatin form, when the DNA is in that form, you cannot see it. But when it gets time to condense to go through cell division, the chromatin is going to start winding around these nucleosomes. Okay, so it's going to wind around the histones. A bunch of histones are going to wind around together to make nucleosomes. Those nucleosome little bundles are going to form coils. Then those coils are going to form supercoils, and then it starts making this huge structure called a chromosome. And when we're looking at chromosomes, oftentimes we'll just draw an X, okay, and so that's going to be a chromosome. But we cannot forget that before the cell divides, it needs to actually replicate. And so when you have one molecule of DNA, it'll go through and it will do replication. So now that we have 2 copies of the same DNA, so everything should be identical. And when it starts condensing in on itself, then it's going to condense. So this is going to be 1 copy over here. This is the 2nd copy. And so how we label things is going to be very important. So one side here is going to be a sister chromatid. The other side is a sister chromatid. It's held together in the middle by this huge centromere. And together, the whole thing is called a duplicated chromosome. Sometimes it's just called chromosomes in itself, but we'll try to not confuse ourselves. But a duplicated chromosome is when we actually see the X. So a duplicated chromosome made up of 2 sister chromatids. When they separate in cell division, and we have 2 cells that are going to have their own copy of the DNA, those sister chromatids are going to become individual chromosomes, sometimes single chromosomes. People have different names for them. So when they become separated, the sister chromatids are called chromosomes in their own right. So sometimes single chromosomes are individual chromosomes. And that is it. So if you have any questions, feel free to go back and re-watch, re-listen. Have a great day, make good choices, and Jesus loves you.